Okay, so the last lecture ended with 7.2 covering the steps of glycolysis, which happened in the cytoplasm around the mitochondria in the cytosol. We're going to look at what's going on inside the mitochondria to finish cellular respiration. The final reactions do occur inside the mitochondria. The one we're going to focus on first is the preparatory reaction. This is also referred to as the acetyl-CoA step. These reactions occur in the matrix of the mitochondria. They produce the molecule that enters the citric acid cycle. And what happens are the 3 carbon pyruvates get converted to a 2-carbon acetyl group in this process. The acetyl group becomes attached to a coenzyme A to become acetyl-CoA, and the carbon dioxide is produced as a waste product. The hydrogen atoms are removed from the pyruvate and picked up to form NADH. And this reaction occurs twice per glucose. I'm going to show you a simplified diagram in just a minute. But to look at the reaction like your book has it, we have the 2-pyruvate and 2 coenzyme A, and NAD plus is going to be reduced, the 2 of them, by picking up hydrogens, and that's going to form 2 acetyl groups with coenzyme A on them, and carbon dioxide as the byproduct, which is what you breathe out. And this is something that you're very familiar with. You've been taught from a young age that you take in oxygen and you give off carbon dioxide. This is why you give off carbon dioxide, because of this preparatory reaction that occurs inside the mitochondria. Let's take a look at this simplified picture that kind of puts things in perspective. So we're in the cytosol and the cytoplasm outside of the mitochondria. Here's the pyruvate. These 3 C's came from the original glucose molecule. So what you're going to do is this pyruvate is going to be taken into your mitochondria, and carbon dioxide is given off as a waste product. So this whole group right here is given off. Then we have the reduction of NAD plus and the attachment of coenzyme A. So these 2 C's here are part of the original glucose molecule. One of them has been given off as carbon dioxide. So we have 2 original carbons here from the original glucose molecule with this acetyl group, and coenzyme A is attached to it. Now remember that there were 2 G3P molecules produced from 1 glucose molecule, and this was during the energy investment steps in glycolysis. So in the energy harvesting steps, those G3Ps got converted to pyruvate, and there were 2 pyruvates for every 1 glucose. So in order for this to cycle through for 1 glucose, it has to go through 2 times, because there are 2 pyruvates for every 1 glucose that we started off with. So the completion of the preparatory reactions is going to lead us into the citric acid cycle, or the Krebs cycle. This is going to occur in the matrix of the mitochondria. We're going to start with a 2-carbon acetyl group that comes from being a product of the preparatory reaction. It's going to combine with a C4 molecule and produce a C6 citrate, a citric acid like you find in oranges and lemons and things like that. Oxidation of the citrate is going to happen by removing the hydrogen atoms. Each of the 2 carbon acetyl groups oxidize to 2 carbon dioxide molecules. There's another waste product of carbon dioxide. This is going to produce 3 NADH and H+, and 1 FADH2 molecule, and also produces 1 ATP by substrate level synthesis. This cycle is going to turn twice per glucose molecule. I'll show you a diagram in just a moment to see why it has to happen twice. And here we have our inputs with the citric acid cycle: 2 acetyl groups, 6 NAD+, 2 FADs, 2 ADPs, and 2 phosphates. Our outputs then are going to become 2 carbon dioxide molecules, 6 NADHs and H+, 2 FADH2s, and 2 ATPs. One important thing to keep in mind is that the citric acid cycle, or the Krebs cycle, is not going to go without enzymes. So enzyme catalyzed reactions occur all throughout the Krebs cycle to form the citric acid. So keep that in mind. So if we kind of look at the citric acid cycle from the grand scheme of things, what's going on, we've already gone through glycolysis and the preparatory reactions, and we are now into the matrix where the citric acid cycle is occurring. So in step 1, the C2 acetyl group that has the acetyl-CoA on it is going to get rid of its acetyl-CoA. It's going to combine with a C4 molecule. This is called oxaloacetic acid, and it's going to combine using enzymes to speed this up into a citrate, or citric acid, which is a 6-carbon molecule again. That 6-carbon molecule is then going to be turned into a 5-carbon molecule, and in the process, one of those carbons is cleaved off, is what we call it, cut off or cleaved off, and forms carbon dioxide. And in the process, we have the oxidation reaction that produces the NAD plus and the H plus ion. This happens twice, so you get 2 sets of those. This 5-carbon molecule then gets another carbon cleaved from it. More carbon dioxide is given off, so you're back to a C4 molecule. And in the process, also 1 ATP is produced through ATP synthesis. In addition, FAD is going to have 2 hydrogens hooked to it and become FADH2. Now the NADH and the FADH, the reason we talk about these is because we're going to need to understand their role when it comes to the electron transport chain. They're going to go on to the next stage. So the C4 molecule then is turned back into the C4 molecule that started with the oxaloacetic acid. And this is going to go through again because remember, for every glucose molecule, we produce 2 acetyl-CoAs. And finally, that leads us to the big electron transport chain, which is the money maker when it comes to ATPs. If ATP is our money, it's our big producer. These are located, this occurs and is located inside the cristae of the mitochondria. That's the highly convoluted, folded insides. It's the high energy electrons that enter the system and low energy electrons leave the system. And 2 electrons per NADH and H plus and FADH2 enter the electron transport chain. The electrons are passed through a series of electron carriers. Those are called cytochromes. And each carrier is reduced and then oxidized. As the electrons pass from one carrier to another, the energy is captured and it's stored as hydrogen ion concentration gradient. The oxygen combines with the hydrogen to form water, and then the NAD plus and FAD are recycled to pick up more electrons from glycolysis from the prep reaction and from the citric acid cycle. As we look at the organization of the cristae, the inside folded, convoluted areas of the mitochondria, we notice that the electron carriers are actually located in the cristae. And as the electrons are passed, energy is used to pump hydrogen ions into the intramembrane space of the mitochondrion. As the hydrogen ion moves back to the matrix, energy is released and is captured to form ATP by a synthase complex called ATP synthase. Synthase is an enzyme complex which speeds up the reaction but isn't directly involved in it. This process is called chemiosmosis. So we see a diagram of the electron transport chain, and you'll notice how hydrogen ion gradients are formed by pushing them into the area outside of the matrix of the cristae, and they then are pumped back into the cristae into the matrix through ATP synthase complex, and those hydrogen ions are then used with ADP to form ATP. So, the energy yield. We said that there was a big yield when it came to ATP molecules with the electron transport chain. So there are 4 ATP that are produced by substrate level ATP synthesis. 2 of them come from glycolysis, and 2 of them from the citric acid cycle. Then there are 32 to 34 that are produced by electron transport chain and chemiosmosis. The reason it's 32 to 34 is because some cells have to pay to pump NADH from glycolysis into the mitochondria, so they lose some of them. Let's take a look at this last diagram for this section, and this shows where glycolysis occurred in the cytoplasm of the cell, breaking down glucose and yielding 2 ATPs. Then we go through the prep reactions that are going to lead to the citric acid cycle and get 2 more ATPs produced, so we have a total of 4. The electron transport chain is our big production line here, and you'll notice that when it goes through the electron transport chain, we get 32 to 34 ATP molecules per glucose molecule that we started with. One of the things that I wanted to bring out, if you notice like the NADH and H plus going through the electron transport chain, I can put in 2 and come out with usually 3 times as much, just depending here, but 3 times as much ATP. Put in 2, get 6 ATP, put in 6, get 18 ATP. If you look down here at the FADH2, we put in 2 and we only get 4 ATP, so the amount's only doubled. And the reason behind that is the electrons in the FADH2 are just not as excited as the ones in the NADH, so we don't get as high a production level. However, if this is an efficient cycle and it goes through like it should with oxygen producing carbon dioxide, that glucose molecule should yield us a total of 36 to 38 ATP molecules. In the next section, we're going to look at taking the last part of section 7.3 and 7.4, which is fermentation, and discuss the efficiency of production through the electron transport chain and what happens when a cell has to go through making energy without oxygen being present. So we'll start there with the next section.